The science of genetic engineering is not new to the world, but it is one that is still taking baby steps in Kenya. And it is a science that is widely being marketed as part of the answer in feeding global populations. But even though commercialization of anything and everything that has been genetically modified is currently illegal in Kenya, scientists continue to research the technology and its viability for the Kenyan food system. And the Biosciences for East and Central Africa Hub at the International Livestock Research Institute campus is one center that hosts laboratories where scientists are developing GM crops.
We are trying to develop disease-resistant bananas which will be resistant to bacterial wilt disease, because this disease is really devastating the bananas in Eastern and Central Africa. We started the project in Uganda, where we developed some varieties by inserting the genes for which can provide the resistance to this bacterial disease. So we have now proof of concept that these genes, the two genes which we are using, can provide resistance to bacterial disease.
To genetically modify a crop simply means introducing a gene or DNA, oftentimes from bacteria or a different species, in order to make the crop immune to pests, disease, or herbicides. And to do this, the genes are carefully studied in the lab, isolated, extracted, and then transferred into the intended crop.
Improving crops include a number of areas. One is to look at the genes that confer specific phenotypes, and you can characterize them, you can select them to be more present in some populations, and that's linked to breeding.
We have inoculated the bacteria in these plants, so for the artificial inoculation, and now these are under observation.
And the modification doesn't take a day. Take potato for example. The International Potato Center at the Baker Ill Rehab is working on a GM potato which is resistant to let blight disease and is ready for field trials. But to get it from the lab to the field is a process that takes years.
There have been efforts before trying to transfer these genes by conventional breeding, but it took a lot of time, about 46 years. So using genetic modification, we are able to reduce the amount of time needed to get these resistant genes from the wild potato into the cultivated potato to about 9 to 12 years.
This is a greenhouse at the Baker Ill Rehab, whereby all the crop that has been developed in the lab is tested. And because these are controlled conditions, the noise you hear is also part of the controlled conditions. Now this is also where the crop that is genetically modified is tested, and we have 3 compartments with GM crop. The first compartment has GM banana, the second one has cassava, and potatoes are at the end. Now all this crop has not gone to field trial stage yet, although the scientists in the project tell me that for banana the field trials begin next year, while cassava and potato field trials are set to begin this year. And again, because this is genetic modification and this is a controlled site, that means that everything that you see here does not get to leave this greenhouse. That means when the tests are done, the crop is destroyed, and also the air that you breathe in here remains here.
If we need the plant back into the lab, we can initiate them back, but otherwise all these plants will be discarded, so they will be chopped, they will be sterilized, like autoclaved before discarding, and the same thing with the soil.
We'll be going for the Confined field trial this season, October. That will be our first Confined field trial.
Transgenic banana and potato are part of the GMO research going on in the country, and scientists have already developed BT cotton, which is ready for commercialization. Genetically modified maize is currently in the 3rd stage of confined field trials and will be ready for commercialization once the ban is lifted. Zainab Wandati, NTV, for Food Friday.